Hello, this is Lindsay Martinez, and I'm here to talk to you about HIV replication. I'm going to start by telling you some facts about HIV provided by the World Health Organization, also known as WHO. 1. HIV virus targets immune cells. 2. Around 35 million people are infected with HIV globally. 3. Combinations of antiretroviral drugs, also known as ART, are used to slow disease progression. 4. About 3.2 million children are infected with HIV. And 5. HIV is the strongest risk factor for contracting tuberculosis. Now that we know a little bit of information about HIV's global impacts, we can look at the structure of HIV before I go into details about the replication cycle. HIV is an enveloped virus that has a single-stranded RNA genome. The virus also codes for its own enzymes, specifically integrase and reverse transcriptase, and proteases. The viral genome is enclosed in a capsid within the envelope. And two major glycoproteins play a role in the replication cycle: GP120 glycoprotein, which is involved in attachment, and GP41. HIV replication starts when GP120 binds to a CD4 receptor on a host immune cell. CXCR4 or CCR5 chemokine receptors are also important, and we'll see that coming up. When GP120 binds to the CD4 receptor, it causes a conformational change which helps the binding of the co-receptor. Once the co-receptor binds, the virus is brought closer, and fusion will be occurring very soon. CD4 receptors, chemokine receptors, and GP120 disappear, allowing GP41 to insert itself into the host membrane. It can then fold upon itself and pull the membrane of the virus closer and closer to the host membrane until fusion occurs. Once the opening is big enough, the viral capsid can enter into the host cell, where it will degrade in the cytoplasm and release the contents of the capsid. We can see in the illustration that DNA is already being synthesized from the RNA genome, right as the capsid is degrading. Before I go into detail about that process, I want to talk about reverse transcriptase, the enzyme coded by the virus in order to make DNA from its RNA genome. Reverse transcriptase has three main functions. First, it synthesizes single-stranded DNA from viral RNA. Second, it degrades that RNA. Third, it synthesizes new DNA from the DNA template and creates double-stranded DNA. Reverse transcriptase has a few different areas, as we can see in the illustration: the P66 site, P51, and RNase H active site. RNA begins by traveling through the P66 active site, where the new template DNA strand is synthesized. The RNA then travels through the RNase H active site, where it is degraded. The newly synthesized DNA does not travel there, because we obviously don't want that degraded. Once the DNA strand has been synthesized, it then travels back through the P66 site, and a double-stranded DNA molecule is created. Let's look into a little bit more detail about how the first single-stranded DNA molecule can be synthesized by reverse transcriptase. Reverse transcriptase cannot just bind to the viral RNA. It needs a tRNA primer from the host. It can then synthesize the new single-stranded DNA molecule, which will end up being larger than the original viral RNA because of duplications of the long terminal repeats. Once the single-stranded DNA is synthesized, we can see again that the double-stranded DNA is formed by reverse transcriptase. Once double-stranded DNA has formed, a pre-integration complex associates with the DNA and helps in the transportation to the nucleus. It travels through the nuclear pores where it then cuts the newly synthesized double-stranded DNA and forms sticky ends. Sticky ends will help with the integration of the viral genome into the host double-stranded DNA. Once the viral genome has been incorporated into the host genome, we can now call it a provirus. The TAT-TAR complex that is associated with RNA polymerase II binds to the proviral DNA and can synthesize mRNA. Now we're going to be talking about translation. Depending on the type of protein that will be produced in the end, mRNA can go in two different paths. It can travel into the cytoplasm and be translated by ribosomes there, or it can travel to the ER where it can be translated. If it travels into the cytoplasm, polyproteins are spliced and migrate towards the cell surface where they eventually are packaged into new virions. If it travels to the ER, after it is translated, the proteins are processed and packaged and released from the Golgi apparatus and vesicles. Once all the viral proteins are assembled, they will travel to the cell surface where budding will occur. HIV follows a type 2 budding mechanism. That means that matrix proteins assemble at the cell surface with the glycoproteins sticking outside. The capsid usually will form around the viral genome, and eventually the new virion will be released. It matures once outside the cell and is like the original virus that we saw in the beginning. Thank you for watching, and I hope you enjoyed this short description of how HIV replicates.